Hello, test. Okay, works. All right, let's get started. Good morning, everyone. Thank you for coming to the Sizing Nanoparticles Satellite Workshop. My name is Jeff Cloxton. I will be your moderator today. I would like to first introduce our first speaker, Dr. Adriele Prima Mello. He comes from Trinity College, Dublin.
Thank you. Thank you very much, Chairman. And ladies and gentlemen, it's a pleasure to give this opening session, and I can see that the night wasn't too heavy on you last night. So I have a very limited amount of time to present because I need to give extra time to Jeff because he has more slides than I do. So going straight into my perspective, the title of my talk is The Experience of Work Within the EUNCL and Some Consideration on a Project that is going to be going forward. So the main key point that I want to sum up of what we've seen in the past day and a half here at CLINAM is the fact that we are facing the so-called translational value debt, where technology platform and technology with a research level around 3 fail to actually make it into the actual candidate with nanotechnology encapsulated. And what we have done historically and through looking through literature, there's a success rate to move from the first compartment of development and optimization where the research and academia are involved that goes down. But the key point is 30% of the nanotechnology-enabled products are failing at the early phases. So this is a given. And most of them, they fail in physical chemical characterization. And this is one of the aspects that we want to bring forward from the aspect of what the nanomedicine characterization laboratory is also. And on this, we will come to the next slide. So what we're going to be focused on, this is what Jeff and I started almost a year ago, the aspect of talking about sizing and the need for sizing, which is need in the early stage of development of any nanotechnology products, because the identification of the proper tools can actually also create a clear pathway for regulatory and for metrology, but also can determine the opportunity to have the right tool to not repeat measurements that are unnecessary. So therefore, it can become also a cost-effective solution. And in this chart, which is going to be published very soon as a joint paper between some of the EUNCL partners, we actually put in that the key measurement that is accepted by the regulator as the electron microscopy diameter could also be combined or collaterally united with dynamic light scattering, which we all know is DLS, which is a light technique, particle tracking analysis for multimodality, resistance pulse sensing, which is also an electrical resistance measurement, and resonant buoyant mass, which is a new technique that is coming on the horizon. I'm going to dwell a little bit into the detail, but just to let you know that there's a pool of orthogonal technologies that came from the early stage where only DLS was the only characterization tool for the early stages. Who is EUNCL? EUNCL is a multi-partners pan-European project, and I'm very fortunate to be sitting in an island, but also very well connected across Europe, where we offer support to partners that they want to develop nanomedicine or they are early stage, and we help them to overcome the so-called translational value of debt. And therefore, we help them to make it to the actual preclinical successful data set, or we support them into their problems. The website is euncl.eu, and we are free of charge as initial consultation. And there's going to be a full session, which is going to be tomorrow, is session 34, which is going to be all dedicated to EUNCL, and every detail will be then described by some of my colleagues in the audience. But also we're going to have a discussion around this this afternoon, and there's posters as well, if you're interested to learn more about the session. So what we are, therefore, we are open and facilitating the so-called translation, the early phase of translation, and any successful application from the perspective of submission will actually get all our full attention to support the so-called accelerated translation. So we're going to enable till the TRL 5, which is the documents that are required to go into a clinical investigation. Into my experience, why I come to EUNCL, just briefly, because I work in Translational Medicine Institute, where I work directly with clinical research and hospital facilities. So we actually have clinical trials ongoing. We are a laboratory for biological characterization of advanced materials. So we do from graphene to carbon nanotube to toxicology studies. So we have a wide span of knowledge. We're involved in several project, but we also patient-oriented. With one of my colleagues, I was just discussing about ICU solution for immediate rapid detection, for instance. So therefore, the vision of our institute is very simple: translating novel discovery from bench to bedside. Nothing more complex than that. We need pharmacodynamics, we need toxicology to do into proof of concept. This is at the basis of our institute. So therefore, that's why we also fit within the European nanomedicine characterization, because our medical need and the unmet clinical need fits within the need for new advanced therapeutics and advanced treatment. So to present you the basic slide, but you will find it in the paper that we just recently published, and I'm delighted to share this success with Peter Wick from EMPA. We actually collect all the latest and the most advanced technologies that are available in the sizes as instrumentation. This, 10 years ago, was only reduced to Zetasizer and DLS. Now we have a full selection, and we are very happy that in this audience there's going to be people that are going to present. A Cordovan technology is going to present about the Vascoflex. And I'm very happy that Jacob is here, so he's going to present in the afternoon his latest offline technology and online sizing, which is very exciting because you go outside the actual complexity of the analytical characterization. We have NanoSite, which is established as ASTM and ISO, so they're finally certified standards that can actually comply for multiple polydispersity. So therefore, you can look at complex particle characterization in different solvent. Or you can actually go in more complex, like the ViewSizer, which is a 3-laser system where you can actually have multiple tracking analysis in a wider size range characterization. Further into, I'm not going to dwell too much because Jeff has promised to impress us with all his slides. So it has some application, especially with the Spectrodyne. But what is important to know is that we go from the standard sizing. We actually now can combine concentration measurement, which is another mandatory requirement in a dossier preparation, because otherwise you cannot couple your carrier concentration versus your active principal ingredient. And when you do this relationship, you cannot do the drug loading versus the drug release calculation. So it's quite important to have one tool that can catch both. And this is why we're moving forward. So DLS, everybody knows what is DLS. So therefore, we have a sample is detecting through two detectors, two different angles. The detector records the fluctuation. The fluctuation is actually done by the shift of the diffusion, and this is detect the peaks. And in this case, we detect in a 60 and a 200 nanometer particle, and you can see that you can calculate the hydrodynamic based on the diffusion coefficient. So in essence, but more detail can be found into the technique. Nanoparticle tracking analysis, in comparison, has a particle suspension in a liquid. The laser beam comes at an angle, and through this one, we're looking at the scattering or the shining or the particles. So therefore, the optical density of the particle gets caught in a planar view. And through this one, we're recording an image. By recording the video, we actually have captured the particle number in the size view. And through this one, we calculate the diffusion equation by Stokes-Einstein equation, and we can actually identify peaks, and these peaks correlate to the concentration and the distribution. And in this case, we have a 100 nanometer particle as a calibrating particle, as you see how sharp it is. Where we actually did the most of the analysis as the experience of EUNCL, we look at the cause-effective analysis, where we actually have the implication and the factor that are influencing a good measurement. Because it's one thing to have a tool, is another thing to know how to use it, and have protocols that actually enable you to identify characterization in particle sizes and concentration within standard and within industrial standard. And what we have identified in this particular diagram for DLS, the causal effective, and this is with the help of Matthias Röslin, who has been interacting with NIST for several years, where he actually created this fishbone identified. The red point are the one that I'm going to highlight your interest, which are not common to any other technique, where the temperature is always one critical parameter, because temperature can actually modify your readout in sizes and in concentration. Another aspect is you actually have to tweak and adjust your system depending on the viscosity of your solution, depending on the concentration of your sample. So you actually have to make adjustment while you do the measurement. And this here we reported all the critical parameters, because it's important that even the regulatory and industry knows what are the factors that are getting into the equation for a proper characterization. When we go to NTA, as you can see, it's far simplified because we don't have a record of the scattering light, but we have an extra branch where we actually have to have a dynamic detection because we're recording videos. So therefore, we've gone from a static picture, from detecting diffusion, to a picture where we record 6 videos in a complete sequence of a measurement. And as you can see, the chart is far simplified, despite size, shape are 2 key critical parameters, because we are looking at the diffusion and the scattering on a plane. So therefore, the shape and isotropy can actually become an issue. But I'll throw this question to Jeff, because Jeff is better than me on this. So when NTA, as we said, allows you through software, through multimodal detection to identify multi-peaks, here we have 100 nanometer, 200 nanometer, and 400 nanometer nanoparticle. With DLS, you actually have a simple approximation, which is only done by the bell-shape detection. Here you can actually identify how the fitting of the software allow you to have multi-peaks detection for polydispersity. When we go to DLS, on the other hand, we can do nonlinear fitting, where we can actually identify the peaks of the measurement in complex solution. In this case, we use fetal calf serum. So therefore, it's a more complex and is a more sticky environment compared to actually the ideal DI water. MANTA, from the other side, has by using the 3 laser, actually has the depth for the selection of the different peaks based on the wavelength that I'm using for the laser. So therefore, you can actually identify the polydispersity by the 3 sensitivity range of the 3 laser. So bring everything back. Obviously, I presented the analytical part, but in a journey on an oncological nanomedicine, the most common is the complexity of the interplay. We have to cross barriers, as there is a special session for barriers. We also have to take into account that most of the time is in intravenous administration, so there's a relationship that is established between the ideal particle and the biological physiological fluid where we're interacting. So there's the so-called formation of protein absorption, there's corona forming, so we need to take into account. And through this analysis, we can see that we are moving towards advanced characterization from the analytical perspective. So what I'm cutting to the end of my presentation, I presented you a number of characterization techniques that brings you beyond the sizing only, but to the concentration aspect, which is critical on the determination of what is going to be your effective nanomedicine for translational pathway. We have identified the key parameter as uncertainties in the measurement to outline the technique that allow the user to get better understanding on the measurement you're doing. One point is to have a calibration particle that you can use to calibrate. The other one is to know what parameters are influencing your measurement. If you're working with a virus, you need to have temperature raising gradient where you do your measurement to look at the activation of your antiviral particle. In another case, you can actually monitor and identify the release of the particle. We have elaborated on the most appropriate characterization techniques, where in the big table that I presented you, which is available online, we have presented the size range that each technique can cover. We presented the sensitivity, the limit of detection. So there's a wealth of information that is advantageous for who has to make a decision on what tool is the most adequate for the development of the nanomedicine. So this is also an aspect that we do within EUNCL. We not only carried out analytical characterization and characterization for translation, we also look into developing new techniques and new protocols. This fits within another project where Patrick Boisseau here in the audience is the coordinator, where we develop in a regulatory science framework. With the new technologies, we need to also develop a science-based evidence where we actually can support the regulatory decision and the acceptance of the nanomedicine development. And this is not done in isolation because the consultation with the REFINE project is actually shared across the different organizations that are involved in the creation of standard, into the dialogue of the harmonization of the protocols, but also interplay between the regulatory agencies. I'll leave you this one to say we haven't addressed all the complexity that we are experiencing here at CLINAM because there's such a multivariate approaches. But again, I'll bounce this one on the playground of Jeff because he's going to have and is going to present some data that is very exciting on tackling some of the specifics. I'd like to thank everybody that has contributed to this work. I'd like to thank EMPA and Matthias and Peter for their outstanding work that they've done with us on the development of the characterization, my team, and I'll thank you for your attention.
Are there any questions?
In your poster on the DLS fishbone, you have written there concentration. I hope you're not suggesting that people can use DLS to measure concentration. Imply that, surely you wouldn't be that.
No, no. If you see,
Some people think you can, but you clearly
no, if you saw on the table we put concentration, there's a big no.
but on your poster you've got it written up on your poster, the slide, DLS, you had concentration written there.
No, I didn't write concentration on the DLS.
Go back and have a look.
You look at the intensity of the peaks of your light scatter, but you don't do concentration. But I double checked, but I'm pretty sure that they didn't write concentration. We are very well aware about the limitation of the concentration characterization. NTA, particle tracking analysis, allowed you this. Manta with the view sizer, Spectrodyne. Every technique that has a single counting of particle allowed you concentration. DLS doesn't have such a resolution, I'm afraid. I hope I answered your question. Thank you. So we switch.